Hello, welcome back. Let's carry on looking at important organelles in cells and the role they play. Next one we're having a look at is the Golgi Apparatus, and that is important for packaging and secreting substances. And in matric you're going to be learning about hormones, and hormones are produced by the Golgi Apparatus in the gland cells in your body. If we look at this picture of a cell, that is what we're looking at now, the Golgi Apparatus, which is enlarged here. It's flattened stacks of membranes, but the important thing is that those little round things, that is the hormone that has been produced by this whole Golgi apparatus. So once the hormone has been made, all these little sacs, or you could say little bubbles of hormones that have been made or packaged inside the Golgi apparatus. So let's have a look at how this works. This is a slide showing part of the cell. There's the nucleus, and here is the Golgi apparatus. The functioning is linked to what we have just talked about the ribosomes, because the ribosomes actually make the protein. That is then sent, there's the protein there, that is then sent to the Golgi apparatus, and the Golgi apparatus packages it into little blobs. Okay, and these little blobs of hormone are called vesicles, and those vesicles then move to the cell membrane and they open, and the protein moves out into a duct, which then would take it to a different part of the body. So the Golgi apparatus works with the ribosomes to secrete substances. Okay, next thing, and you'll be spending quite a bit of time on this, is revising the nucleus. So this is a model of the structure of the nucleus with the nucleolus, the chromatin, the nuclear envelope, keeping it all together. The fact that there are little openings, which is quite important, because that is how the instructions from the nucleus get out. And right next to the nucleus, we have the endoplasmic reticulum with the ribosomes, because that is where the protein is going to be made. You would probably have seen the nucleus drawn something like this, with the nuclear envelope, the pores, the DNA or chromatin network inside it, and the little blob of RNA, which is called the nucleolus. Now, why is the nucleus so important? And that is because of the 46 pieces of DNA. So if that's our cell, that's the nucleus. What you've seen as chromosomes here are all made of DNA. And on the DNA are the instructions for all the cell activities. Now, you probably are a bit confused, because sometimes when you see a drawing of a cell, the DNA is long, thin threads, which is the chromatin network, and other times you see a drawing of a cell and they're little blobs called chromosomes. Now, what is important is each cell has got all the instructions for doing everything inside your body. But a cell only carries out some of the DNA instructions. For example, brain cells will be carrying out the part of the DNA, the genes on the DNA, which tell it how to be a brain cell, how to make proteins that brain cells need. It won't be worrying about producing enzymes like your stomach cells will be. Now, on the DNA are genes. So this chromosome would have patches of it which are different genes, and the gene is the piece of DNA with instructions for making a particular substance. Example, enzyme. We talked about the stomach producing enzymes. Let's have a look at how chromatin, chromosomes, and DNA all fit together. Okay, first of all, in an ordinary cell, that is what the DNA looks like. It's long, thin threads, and those long, thin threads we then call the chromatin or chromosome network. And you'll be learning next year it's made up of DNA, which has a particular structure. It's called a DNA helix. But then, because the chromatin is very, very thin, during cell division, the chromatin, and you can see here, it's being wrapped around proteins called histones to form larger structures. And that is the larger structure formed, and you've seen pictures of that. That is called a chromosome. And a chromosome is made up of replicated pieces of DNA all wound around each other to make a chromatid. This one here is the other chromatid or copy of the DNA. So what is on these two chromatids are actually the identical DNA. How can it be identical? Just before cell division, the cell replicates. So if that's what the chromosome looks like, after replication, that's what you will see. So that's why sometimes chromosomes are shown as single. It depends, have they replicated or not. Okay, so these copies form the chromatid. They're held together by a centromere. And the important thing is that when the cell divides, this chromatid will go to one new cell, and that chromatid will go to the other new cell. So each new cell will get a full copy of all the instructions that were on the DNA of the original cell. And that's important because those instructions are what is needed for the cell to function. Now, in grade 10, you learned how cells divide in the process of mitosis. So mitosis is what happens when cells need to grow. They need to grow more cells so that you can get bigger. When your nails grow, when your hair grows, etc. And the important thing of mitosis is it produces two identical cells, and they are exact copies of each other. Okay, the first stage of cell division is called prophase. And in prophase, the cell starts preparing. That's what the P is, preparing for cell division. So what's happening here is the chromosomes are starting to form. Remember, before the cell division even starts, before prophase, the DNA has to replicate. So when a cell starts dividing, the DNA is already replicated so that when the chromosomes form, they are double. Another thing that is happening here, and you can see that here, and that is that the centrioles separate. And these blue lines, those are the spindle fibers that the centrioles are busy forming. So what is happening here? The nuclear membrane is disappearing because it would only get in the way. Because the important thing in cell division is that each new cell has to have an exact copy of all of the instructions. So these chromosomes have to be very carefully sorted out, and that's why the cell undergoes all of these stages. Second stage is metaphase, and you can remember that because metaphase is when the chromosomes are in the middle of the cell. The centrioles are at the poles of the cell, which are the ends of the cell, and the spindle fibers have formed, and the spindle fibers have attached to the chromosomes. Third stage, anaphase, and you can think of away, A for away, because that is when the chromosomes split and the two halves of each chromosome move away from each other. So that this cell on this side is getting one copy of the instructions, all it needs, and this cell on this side is also getting a copy of the instructions. Once the chromosomes arrive at the poles, that is the last stage, or telophase, and each new cell, or new cell that's going to be, now has a complete copy of all of the instructions, and now the cell can start dividing in cytokinesis. You will be doing meiosis in grade 12, so you need to remember how mitosis occurs. It'll make it easier for you when you get there. There's interphase, then there are the 4 stages of nuclear division, then cytokinesis, and this is what you can use to help you remember. IPMATCH. I for interphase, when DNA replicates. P for prophase, when the chromosomes form. M for metaphase, when the chromosomes are along the middle of the cell. A for anaphase, when the chromosomes split and move away from each other. And T for telophase, when the nuclei reform, followed by cytokinesis, when the cytoplasm divides. I hope this will help you when you get to your life sciences classes next year. Good luck.